Hey everyone, I hope you're doing well. I wanted to go through a few things with you today about the cancer detection lab using gel electrophoresis. This is a lab that I've used many times in my classroom, specifically my AP Bio course. But I do think that this is a great lab that can be used in an anatomy phys course, in an underlevel biology course, or a biotech course. So this lab has a lot of applications, and hopefully you can find something that will fit the niche of what you're looking for in your particular class. So the overview of this lab basically looks at a simulated family, and the kids are going to end up making a pedigree of this simulated family and looking to see which individuals are at risk for a certain type of inherited form of cancer. And then they're going to go and they're going to do gel electrophoresis on those individuals that are at risk and see if they've actually acquired the mutation for the inherited form of cancer. The specific type of cancer that we're going to be looking at is a type of colorectal cancer, and it's also known as Lynch syndrome. There are 4 caretaker genes that kind of control whether or not individuals are predisposed to get this type of cancer. And for me in particular, the genetic portion of this and also the biotech portion, which we'll get into in a minute, is really what I focus on. But I think this is also a great opportunity if you're teaching an anatomy phys course, or even if you have a unit on some systems, to work biotechnology into that area. So I do think that this would be a great time if you are teaching the anatomy of the digestive system to really go through and kind of revisit maybe some of the things that you've learned about the digestive system and even different tissue types. Because as you'll see in a moment, when we look at the inheritance pattern, it also really does depend whether someone will develop this type of cancer, that this is really happening in epithelial tissue that lines the colon. So the gene that we're going to be looking at, even though there's 4 caretaker genes, 90% of the mutations for this type of cancer happen in 2 of them, the MLH1 and the MSH2. And this particular lab looks at the MSH2 gene, which is located on chromosome number 2. And this is a specific type of caretaker gene that's involved in DNA mismatch repair. And that leads me to my second point. This is the focus that I usually have with my class in my AP Bio class. I think kids a lot of the time when they're in an underlevel biology course, there's a lot of really good information that they already learn in mitosis labs that they do. And so I was really looking to take it to the next step and try to incorporate a lab that looked at cell signaling and also the cell cycle. Because that was, not only is it something that they didn't do in earlier years, but it's also a place in my curriculum where I had some holes where I didn't have a lot of labs to really incorporate. It was more just kind of drawings, textbook work, looking at stuff under the microscope. So 2 things that I can really focus on here are what is the role of a tumor suppressor gene and what is the role of proto-oncogenes in the development of cancer. So this MSH2 gene is a type of tumor suppressor gene, and it's involved in DNA mismatch repair, which pretty much means that it basically corrects DNA errors in DNA replication. And it's a tumor suppressor gene because overall tumor suppressor genes slow down the cell throughout the cell cycle and basically makes them, the cells stop at certain checkpoints to make sure that everything is going okay in the life of the cell. So is the cell healthy? Has the DNA replicated properly? Even when it comes time for mitosis, have all of the spindle fibers attached to the chromosomes properly? So this is not, without getting into too much detail, this is what I focus on in my class instead of some of the other aspects of really like the digestive system and things like that. So once we've kind of gone over some of that stuff, the first hands-on thing that the kids do in class is they build a pedigree for a simulated family. And this particular cancer is inherited in an autosomal dominant fashion. So the individuals are really only need 1 copy of the gene to be predisposed for this cancer. And one point of confusion that kind of comes up with this is that really to develop this cancer, an individual needs 2 mutated copies of the gene, which we all know is characteristic of a recessive pattern of inheritance. However, because the cells of the digestive system, specifically of the colon, are replicating so fast because they're always sloughed off into the gut, if an individual has inherited one mutated form of the gene, the chances are that the other one is going to mutate is pretty highly likely. And that's why they consider it pretty much an autosomal dominant inheritance pattern. So that is another thing, if you're teaching an anatomy phys class, that maybe you want to bring back in about the lining of all your different systems and the epithelial tissue and why all of those cells are replicating so fast in this specific area. So once the kids have built this pedigree, so they're building it, then they are going to identify specific individuals that are at risk for this cancer gene. And when they do that, one thing from a biotechnology standpoint that I really, really like about this lab is it's very simple in the fact that the only biotechnology technique that they're using is gel electrophoresis. So this lab has pre-digested samples. So what that means, pretty much if you were going to genetically test someone, you would gather their cells and do a DNA extraction. And in that DNA extraction, you would gather up their entire genome pretty much. And in a normal testing situation, once you've done that DNA extraction, you would go through the process of PCR. And PCR, which we'll talk about through the Baxter Box at Home program through some other labs, basically isolates a very specific gene. And in this case, for us, it would be the MSH2 gene. And then it makes many copies of it. And after we've done that, then we would go into a restriction digest. And what a restriction digest is we would use a specific enzyme. In this case, the name of the enzyme is APO1. And what a restriction enzyme does is it looks in your DNA sequence to see if there's a specific sequence called a recognition sequence that it will cut. And in this case of our MSH2 gene, if it happens to be mutated, our restriction enzyme is going to recognize that mutation, and then it's actually going to cut the gene at that site. So luckily for us, with this particular restriction enzyme, the APO1, the mutation introduces a restriction site for this enzyme. So what happens is, if you are an individual that does not have a mutation, when your DNA is treated with this restriction enzyme, your DNA will not be cut. If your DNA does have the mutation, when it's treated with this restriction enzyme, it will be cut into 2 pieces. Now, in this particular lab, all of that has already been done for us. So this is a great lab to introduce biotechnology into your classroom, because this could be something that you talk about, but the kids aren't actually doing this quite yet, but they will do the step of gel electrophoresis. And then in later labs throughout the year, you can start to work in certain restriction enzyme labs, and then also labs that encompass and incorporate the process of PCR. So for me personally, this is the first gel electrophoresis lab of 5 that I do in my AP biology class. Now, one thing that you have to remind the kids, and for me, we haven't gone through genetics yet, so we're kind of like learning how to do this pedigree. And I'm trying to remind the kids that you get 2 genes from every parent, right? One from mom, one from dad. So you have 2 alleles. We really have to talk about, if you're an individual that is homozygous, normal, so you do not have a mutation, you have 2 copies of this gene that is not going to get cut. And if you're homozygous mutation, which is pretty rare, then you are going to have 2 copies of this DNA sequence, both of which are going to get cut. And if you're heterozygous, then you're going to have 1 of each. And pretty much because this sequence has been isolated through the process of PCR, it is a certain size. And so the 2 fragments here should pretty much equal the size of this 1 fragment that has not been cut with our restriction enzyme. So what happens when we look, So what happens when we look at our results here? If you're an individual, so we're going to load our gel here, and in the first lane, we're going to have a standard. So these are going to be DNA bands of known sizes. And then we're going to have one lane of an individual that is not affected. So they do not have the mutation for this cancer type. And that individual then, as we know from this previous slide, is going to have 2 copies of this gene that do not get cut. So what that is going to look like on our band or on our gel is just going to be one band. Now, they really have 2 copies of this gene, but they're just overlapping one another. So you don't see those 2 banding patterns. Okay. If you're an individual in lane 3, this is going to be from a tumor sample. So that means that that is an individual that had 2 copies of this gene. And so each copy is going to get cut. So you're going to really have 2 copies of this fragment and 2 copies of this fragment. So on our gel here, that's going to look as 2 bands. And then if you're an individual that's heterozygous, you are going to have one gene that was not cut and one gene that was cut with our restriction enzyme. So that will end up looking like this is the gene that was not mutated and was normal. And then these 2 bands come from our mutated gene that was digested with our restriction enzyme. So the kids they're going to need to know how to interpret their results. But for the most part, what they're going to be doing in this lab is taking a bunch of samples. They're just going to load the samples in their wells here. And because DNA is negative, we're going to hook up this gel to electrodes, and the negative end of the gel is going to be on this side, and the DNA fragments are going to move through our gel towards the positive pole, with the smaller fragments moving faster in a given period of time than our larger fragments. But I'll go over some of that in the lab demo that I did for you in a different video. Just to show you what my kids came up with, because I know sometimes we're always curious about, well, okay, so we do this in class. What does this really look like when the kids do it? These were 2 different gels that my kids did. And you can see here that we got pretty good results. So we've got our standard in this first lane, and I think there were 14 samples. So there was a control sample of normal DNA. There's our one band, like we discussed. Then there was a control where tumor DNA where it was digested. So we have our 2 bands there. The kit also gives you a control heterozygous. So you see your 3 bands there. And then here are all these different individuals, and I put their names in, but you'll see when you go through the manual, these are the simulated names from that family. And so then the kids can start to fill in their pedigree with these individuals on what their genotypes are, and if that matches the inheritance pattern that they thought that they were going to get from the pedigree that they made previously. So this lab is pretty reliable. I've gotten pretty good results almost every year, but also I suggest once you do a lab and you get really good results, make sure you save pictures in case if your kids screw it up, because then worst case scenario, you can always just show them pictures from previous years, right? Okay. The only other thing that I wanted to focus on is here's a list of all the at-home labs that we talked about in the orientation and in our Monday meetings. But in case of this particular storyline or this lab doesn't really fit what you're looking for in your class, but you kind of like the idea of having a simple biotechnology lab to do that doesn't have a PCR involved and doesn't have a restriction digest, just to start your kids off. 2 of the other labs that we offer one is a DNA damage lab, where the kids basically are going to expose different DNA samples to light sources and see if they can induce a mutation into their sample. And then also our sickle cell lab, which comes from the company Mini PCR. And if you like the sickle cell storyline, which I'm sure almost all of us, if you're teaching a biology class, are going to talk about sickle cell, basically you are looking at a family again and looking to see if individuals are carriers for the sickle cell gene or not. But that lab also does not involve PCR or a restriction digest in the actual technique of it. Okay, well, I hope that was helpful. I don't want to make the video too long for you, but I will be available for any questions that you may have about this lab if you want to talk about more of these ideas in depth. So thank you very much, and I will see you soon.